Now we have Dr. Niti Puri with us. Ma'am, are you there?
Yeah, I'm here. Hi, you can see me?
Hi. Good
Yeah,
morning, ma'am.
good morning. Yeah.
Okay, so Dr. Niti Puri joined the School of Life Sciences, Jawaharlal Nehru University, Delhi, as an assistant professor in immunology in 2008. And ma'am also has a postdoctoral experience at National Institute of Immunology, Delhi, and as a Fogarty Fellow and a visiting fellow employee at National Cancer Institute, National Institute of Health, USA. During a total of more than 23 years of her service after PhD, ma'am has carried out extensive research in the field of cellular and molecular immunology with special emphasis on pathogen interactions with host innate immune cells. Ma'am has several competitive research projects with collaborations nationally and internationally. Her research findings have been published in high impact peer reviewed journals and presented in various national and international conferences. We welcome you, ma'am. Ma'am's topic for today's talk is going to be multipronged mast cell effector responses and their evasion, facilitating pathogen clearance or persistence.
Okay. So, are you able to see my slides?
Yes, ma'am.
Okay. So, thanks a lot, Namisha, for the wonderful introduction. And I would also like to take this opportunity to thank the organizers as well as especially Dr. Asmita Das for contacting me to deliver this talk. So, and I have been attending the talks also. It has been a very nice session. So, so after such a nice talk about COVID, so this is slightly different because this is what we have been doing before COVID and we still continue to do. So we are also looking at the immune responses, but I will be showing you innate immune responses against two pathogens which are also very prevalent in India, that is Leishmania as well as Mycobacterium, and what our findings have been regarding them and their interactions with the innate immune system. So, okay, so our lab has been studying the role of innate immune cells and especially mast cells when they interact, because these are actually the sentinels of the immune system, and they are the first ones to interact with many, pathogens as well as environmental, you can say, allergens and pollutants, so many certain things. So they interact with them, and they are the ones they recognize, and then they categorize them, and they tell how adaptive immunity should actually counter this. So they are very important. They're signaling the changes that happen in them, their secretion of mediators. They all play an important role in driving the adaptive immunity, which people have given a lot of importance to, especially, the helper T cells, the secretion of cytokines, or the secretion of antibodies. But all that is possible only because initially these innate immune cells actually recognize the pathogens, respond to them, categorize them. They may even take them up and then present their antigens to activate the T cells. So just as immune system has developed, many ways to counter these various pathogens, same way what happens is that pathogens also over time, they are also developing new and new strategies to evade these effector responses. So what I will be showing you today is how this tug of war goes on with it, between the innate immunity as well as, these pathogens, and how we actually need to overcome that to find good therapies and also to get good immunity to many of these infections. So, so you all must have heard about mast cells, but mostly people think of mast cells because they are involved in allergies. So what happens is that we know that they are packed with these granules which contain histamine and many other substances like inflammatory TNF alpha and many enzymes. They are all present there, and when they are activated, usually with an allergen and involvement of IgE, they immediately degranulate, causing release of these nasty mediators, which then give us, severe allergic responses. So for a long time, people thought that that is the role of mast cells. But so far, no human being has been found to lack mast cells. So this shows that they are really important for our immunity. So it was only in the last, you can say, couple of decades that people have started thinking of mast cells as important innate immune cells. And what they have found is that now they know that mast cells actually they are present in the first line of our defense. That means just where the pathogens enter, and they contain many of the receptors which are important for detecting these pathogens. They are known to respond to all kinds of pathogens, be it bacteria, be it parasites, fungi, to all of them. They express toll-like receptors which recognize certain patterns in these pathogens. And then they respond, not just by secretion of mediators, but now people know that directly they can even phagocytose some of the pathogens. They can trap some of the pathogens in making some, traps out of their DNA. So this is something that is called formation of, death by etosis, and leading to formation of extracellular traps, so which then traps the pathogens, and that way pathogen cannot escape and dies right there. And in addition to that, they also secrete many cytokines, which activate adaptive immunity, which help dendritic cells to, generate helper T cells. And also in innate immunity also, that means they actually help in recruitment of other innate immune cells like neutrophils and eosinophils, because of secretion of many of the uh, chemokines and other cytokines. They also cause inflammation of the endothelium. So that is how these neutrophils are then, they know where to come to the site of infection. So these have been shown with many pathogens. And so why is there these, why are these cells important? Because as I told, that they are present in periphery. Usually there is no particular organ, you can say specifically they do not reside in any specific like spleen or any such organ, but they are present all over the body and spread all over the body, where in skin, or in lungs, mucosal surfaces, real near the connective tissue, that is near the blood vessels. So at all these places, all the mast cells, they reside. So usually they are present in the same places where we have dendritic cells and macrophages. So, and they have this ability to secrete certain mediators immediately within, minutes of, because these mediators are actually pre-synthesized and stored in their granules. They are granular cells. And one of the mediators, very important mediators, is actually TNF alpha. So mast cells are the only immune cell known which have pre-stored TNF alpha. And they, because of secretion of many cytokines, they can also, or chemokines, they can also recruit neutrophils and eosinophils and other cells, macrophages, to the site of infection. And recently people are also finding that, because they are slightly long-lived cells, they also can then respond a second time, and they can modulate the responses. So that is again something there, probably their immunity can be trained, so they can become important in that way as well. So, so in our lab, so there are some roles of mast cells which have been well established, like, how they affect other cells, that now people believe that they are regulators of both innate and adaptive immunity. And they also can actually alter the polarization of T lymphocytes because of the different cytokines that they can secrete. And as far as infections are concerned, we know for sure that they are important for tackling the parasitic infections, or you can say worm infestations, because they have the IgE and then they secrete the mediators that helps in expulsion of the worms. But what about bacteria and, other parasites? So for that, it is still not very clear. So in our lab, two of my students, so Nilofer and now Rahul, so earlier students, so Nilofer has actually finished her work. So she is the one who performed most of this work, and we are continuing this work. And she is the one who studied the role of mast cells in various, with interaction with various pathogens. So objectives, aims of our study were to find whether they play a direct role in clearance of some of these pathogens, or also in efficacy of vaccines, because see, vaccines have to give us long-term immunity. So we want to know whether they actually play a role in that as well. And then what are their killing mechanisms, whether it is by phagocytosis or whether it is by extracellular means. So we ultimately want to find whether, they do act as phagocytes or as non-professional antigen presenting cells, as scavenger cells, and what is their crosstalk with other immune cells. So that is so and the two pathogens that I told you we will be discussing today, Leishmania and Mycobacterium. So both of them are intracellular pathogens, and they are relatively, you can say, not so easy for the immune system to see. They actually utilize the immune system. They reside inside cells. Both of them enter in macrophages, reside there, and utilize the mechanisms there for their own benefit, because they can stay there for long periods of time, divide there. They have developed many evasion strategies to, prevent their killing inside these immune cells. So that is why we were interested in them, because we thought that it is not just macrophages that see these pathogens first, but, mast cells also are present in the same place. So when there is an entry of either Leishmania or Mycobacterium, then mast cells must also be seeing them. So what is their role? So this is something that has been, not very, you can say, well discussed or studied. So that is why we thought we will look into that. So I'm sure all of you are aware of mycobacteria, but so see, we have not been studying Mycobacterium tuberculosis, but we decided to look at actually how they interact with Mycobacterium bovis, that is Bacillus calmette-guerin, which is the only known vaccine for MTB. And the reason being that it is easy to work with, BCG in a mouse model. So infection of mouse with BCG actually mimics exactly the infection of MTB in the human system. So we thought we will utilize that to our advantage. And this will also probably give us some clues as to, if mast cells have any role in not providing, very long-term immunity by BCG, because BCG protects the people who get BCG, but, there is a variation in this protection. So while some people may be protected, but some are not, and children may be protected, adults may not be protected. So we can think of, so people are trying to modify actually the vaccination by BCG to try and see how they can improve the immunity. And recently, a lot of research is going on also about the innate immune responses, because that is what may actually drive the adaptive immune responses. So very little is known about interaction of, so actually nothing is known so far about interaction of BCG with mast cells. Though people have looked at the interaction of BCG with macrophages and dendritic cells, they are still looking at the role of neutrophils also, even that is not clear. But as far as MTB is concerned, very recently people found that, MTB is able to enter mast cells, or you can say is phagocytosed by mast cells. It is also detected by many receptors on mast cells, and mast cells secrete many important mediators after this interaction. But after that, what happens, that is not really clear. So we thought that, these what we study will give us some clues about what is happening here as well. So, and as far as Leishmania is concerned, so Leishmania is responsible for causing leishmaniasis, which is a very serious disease, especially in tropical and subtropical countries, and India being one of them. So a lot of cases of Leishmania are seen in India. And this is because of this protozoan parasite, Leishmania, so which actually is introduced into the humans by bite of this sandfly, which is the actually vector. So it is a vector-borne disease. So when it takes a blood meal, then it introduces this promiscuous form of this pathogen into the bloodstream. And there it is actually taken up by, in the skin when it is there, then it is taken up by resident macrophages, and then macrophages are not able to kill it, and it stays inside macrophages, where then it converts into its other form, that is actually the amastigode. It divides there, and it can even be taken to other organs. And again, these infected macrophages, when they are taken up by this, again, sandfly, when it bites in a blood meal, and then that is how it is transmitted. So it will take it up, then inside the gut of this sandfly, actually this again macrophages, these amastigotes are released, and then they again convert to promistigode form, which is again then the one which is transmitted to the next host. So that is how it spreads. And actually there are many different, you can say, uh, different species of leishmania, and they cause different kind of diseases. So some of them actually cause a milder form, which is called as cutaneous leishmaniasis, where the infection is limited to the skin only, and it forms these lesions, and macrophages also stay here. They may only go to the lymph nodes. So L tropica and L major are the two major forms which cause this cutaneous leishmaniasis, whereas another form, which is the visceral leishmaniasis, that is also called as Kala Hazar. So that is the more, you can say, serious form of disease, and L donovani is the major causative agent. And this is where infected macrophages can actually, they go and infect other organs also, like liver and spleen, and this can lead to severe consequences. So for both these diseases that I told, actually some drugs are available, but as you know that, drugs have to be taken for a long time, they are expensive, and also no vaccines, suitable vaccines are there. So, but there have been, for leishmania, there have been some reports where people have looked at mast cells, and they have shown that mast cells may have a role in clearance of leishmania, because mast cells were shown to be recruited to the site of sandfly bite. And also they were shown to be important for formation of granuloma or recruitment of macrophages. But then again, more than that, there is still a big question mark, because it was not very clear whether mast cells are helping the host or the pathogen, or, how does this work. So now I will come to the study. So we will look at, interaction of mast cells with, so we decided to look at the interaction of mast cells with both these pathogens in the in vitro conditions, to see how mast cells actually respond to them, and what are the effective mechanisms that are employed. So, and whether they are killed intracellularly, extracellularly, or what is happening. So since, as I told you, with BCG and mast cells, there were no studies that had been done. So first we employed the mouse system. What we did was that we introduced BCG into the mouse, into the lungs of the mouse directly by gavage. So that means now we are introducing it as a person will get the MTB infection. In a similar way, it was introduced. And after that, we waited for a few days, and after that, we took the mice were, euthanized, and the lungs were taken, their sections were made, and they were stained with different stains to look for different things. So first thing that was done was acid fast bacterial staining was done. And what we found was, so this was actually the uninfected lung versus the infected lung. So in this, you can see that actually a lot of cells were seen, but we were unable to see any pathogen anymore. So which showed, so we had done this 4 weeks after immunization. So that means a lot of cells are there, a lot of immune cells may have come. There were also granuloma formations, but the pathogens seem to have been cleared now. So that means it was cleared by with these various, you can say, by the immune response. So you can see here, so again by hematoxylin and eosine staining also, same thing was seen, that granuloma formation was seen, and there was a lot of infiltration of various inflammatory cells. So they probably helped in clearance of the pathogen. And when we stained, so to look specifically for mast cells, we actually did toluidine blue staining. So toluidine blue stains the mast cells because it stains the granules in mast cells. So what we were able to see was that in the uninfected lung, so there are fewer mast cells. So you can see that we could see only about 2 mast cells per field. But in case of infected lung, actually the number of mast cells increased by almost 20-fold. So now there were a lot more. So that means mast cells had also come to the infected lung, and probably maybe they would have helped in clearance, but that we still do not know. But they were there, and the infection had been cleared. So after that, to look at the interactions, in vitro, we have used 2 different, you can say, kind of mast cells. So one was, we used actually peritoneal mast cells. So from the peritoneal cavity of the mouse, the cells were actually The cells were taken and then they were, by sorting using some mast cell specific markers like CD117 and CD45R, we actually gated on the mast cells and we were able to get purified peritoneal mast cells. So these, out of the total peritoneal cells, only 2% are mast cells. So that is why this sorting was actually required. And in addition to that, we also have a rodent mast cell line, RBL2H3, which also contains these granules. It is an adherent cell line and it is used extensively to study mast cell biology and also interaction of mast cells with many pathogens. So we utilize that also. So what we saw was that when we co-cultured actually mast cells with both these pathogens, then what was seen was that both the pathogens were actually killed. So, but what we saw was that, so you can see BCG almost 50% killing was seen with mast cells. And same thing, you can see that we studied L. tropica as well as the L. donovani. So that means L. tropica that causes cutaneous leishmaniasis and L. donovani that causes visceral leishmaniasis. So L. tropica was killed to a large extent. So there, the viability was reduced on co-culture with mast cells. But L. donovani was not killed to a great extent. So there was about maybe 15% loss, but not more. So that means L. donovani actually was quite resistant to killing by mast cells. But when we put these two together in transwell, then what we saw was the killing was not there. So that means direct interaction between the two was required. The pathogen and the host was required to kill the pathogen. So that means initially it was not due to some mediator that was being secreted due to which the cells were being, due to which the pathogens were being killed. So further, for leishmania, we also actually tried to see what was the mode of their killing. So we tried to do annexin 5 staining. That is, we looked for apoptosis in leishmania. So a lot of leishmania was actually being killed by apoptosis, as we can see. So there is PS externalization here, that is phosphatidyl serial externalization. So leishmania was killed by apoptosis as well. So further, so since we were seeing the killing, so now we wanted to see whether, the killing was intracellular or extracellular. So to see that, we wanted to see whether these pathogens were being phagocytosed. So we stained these pathogens with CFSC. So that means now the pathogens became labeled. So they are fluorescently tagged. And then we incubated them with our mast cells. And after that, we actually took the mast cells and we looked at their uptake. So by fluorescence microscopy as well as by flow cytometry. So this slide here shows that the label was taken up readily by all the pathogens, and the label was quite stable up to 24 hours. You can see here, so this is actually labeled BCG. You can see that now it is green, so it can be easily followed. So what we found was that these mast cells were able to take up, L. tropica very easily, but not L. donovani. So L. donovani was not taken up at all. So this again showed that L. donovani seemed to be resistant to mast cells or was able to evade phagocytosis by mast cells. And when we treated the mast cells with cytochalasin D, which actually prevents the actin, rearrangements, then we found that the killing was reduced. So here, without cytochalasin D, a lot of L. tropica was being killed. But when we had added cytochalasin D, the killing was reduced. So that means that some part of pathogen killing is because of uptake. So that means it is by intracellular means in case of L. tropica. So same way for BCG, we also wanted to see, what is so not just BCG, but we also wanted to see whether mast cells are able to take up mycobacterium derived proteins also. So for that, we sonicated BCG and then biotinulated that and then studied the uptake of that as well. So you can see here, so this is actually the uptake of CFSC labeled BCG, live BCG. So a lot of BCG was taken up. And especially here also at higher MOI, that is multiplicity of infection, more and more was taken up. And so what was the mechanism? So, mechanism. Pathogen can be taken up by various mechanisms. It can be lipid raft dependent, caveoli dependent, or there can be some signaling involved. Of course, actin polymerization plays a role. So, and also, at least for the professional antigen presenting cells, macropinocytosis also plays an important role. So we wanted to test all these ways by using certain inhibitors for each of these. And what we found was that on using inhibitors, you can see that when we actually treated with methyl cyclodextran, which extracts cholesterol, you can see here by Philippine staining that cholesterol has been extracted from these cells. And after that, when we incubated them with BCG, you can see that there was a tremendous inhibition in uptake, BCG uptake, when we treated them with all these inhibitors. But there was no effect of what menin. So that means PI3 kinase signaling was not involved in this case. But we found that lipid rafts, caveoli, as well as actin rearrangements are involved in uptake of BCG. So, and then as we know that opsonization also plays a role, especially in professional phagocytes. So what we did was we actually raised some serum, or you can say anti-serum to BCG by immunizing mice with BCG and getting the polyclonal sera. So we treated BCG with that so that they are now coated with antibodies, and then we fed them to, or we can say we fed them to mast cells, and then the uptake was enhanced. So you can see almost fourfold enhancement in uptake. So this is what we see with professional APCs like macrophages also. So that means opsonization helps in uptake by mast cells as well. So then we wanted to see that after BCG is taken up, so where does it go? So we stain, so usually the phagosome is supposed to then fuse with the lysosome to to make the phagolysosome where the pathogen is usually killed. So we stained the late endosomes and lysosomes with LAMP3, and then we tried to see whether BCG is able to co-localize with LAMP3. So we did not find much co-localization between the two. So that means that though it is internalized, but it is somehow inhibiting the phagosome lysosome fusion. So this is something that is also true of Mycobacterium tuberculosis. So tuberculosis is MTB is also able to actually evade the immune responses by preventing the phagosome lysosome fusion. So then I told you that macropinocytosis, which is also an active process and involves cytoskeleton, this is the drinking, or you can say by this soluble proteins can be taken up. So mast cells have not been shown to perform that, but we tried with these mycobacterial proteins to see if they can be taken up like that. And what we found was that the sonicated BCG was also readily taken up by mast cells. So you can see here, so this actually we are detecting it with, since this is biotin related, we are detecting it with streptavidin that is linked to FITC, or a green dye. So you can see it is taken up, and this DAPI, this is the nucleus, so it is also internalized. So to see the mechanism, we saw that if this uptake was actin dependent, so it was, because treatment with cytochalasin D actually inhibited this uptake. And also when we performed this uptake at 4 degrees, so this was again inhibited. So this showed that this was an active process. So this is the first report where we saw that mast cells also, like other professional antigen presenting cells, can perform macropinocytosis as well. And when these mast cells were activated, for example, by this providing this calcium ionophore ionomycin, then this uptake, the macropinocytosis, increased manifold. So that means they were able to take up the pathogen even more strongly. So activation is increasing the uptake. So here, if you see, so the comparison of, uptake by these various, you can say, of these various pathogens or pathogen-derived antigens. So important thing is that both this Mycobacterium bovis as well as L. donovani, they are showing some degree of evasion. Actually, L. donovani shows a lot of, you can say, evasion, because it is completely resistant to phagocytosis. It is not taken up by mast cells at all. And M. bovis BCG, though it is taken up, but it is not able to go to the fused with the lysosome. So that means it is inhibiting that. So other than that, L. tropica is, I think, easily taken up and is probably killed inside the mast cells as well. So after that, so the other mechanism of, so we looked at the ROS production, because reactive oxygen species are known to kill most of the pathogens, either intracellularly or also extracellularly. And also mast cells are known to degranulate, and by degranulation also they release many antimicrobial substances which can kill the pathogen outside the cell. And as I told you earlier, that mast cells have also been shown to generate these extracellular traps where they release their chromatin to ensnare the pathogen and also concentrate the various antimicrobial substances to kill the pathogen. So we tried to see whether all these mechanisms operate in these pathogens interaction with mast cells. So we found actually a lot of ROS production in mast cells in interaction with all these, especially with BCG, there was a lot of ROS production. So this we detected by using a dye, which is this CMH2 DCFDA staining. So this actually, due to oxidation with ROS, it gives a color, and that is what we have seen. So, and L-Donovani also caused a lot of, interaction with L-Donovani also caused a lot of ROS production, even though it was resisting phagocytosis, but ROS production was still there. And L-Tropica also caused ROS production in mast cells. But when we co-cultured them in transfers, then again ROS production was not seen. So that means again direct interaction with pathogen is required to get the production of reactive oxygen species. And to see whether RAS production is actually involved in killing, we also treated these mast cells with catalase, so which will quench the reactive oxygen species. And treatment with catalase actually caused, you can say, that it prevented killing of the pathogen. So initially there was a lot of killing, but in presence of catalase, now more cells were, or more pathogens were alive. So this was seen for both L. tropica as well as for L. donovani. So this implicates ROS in parasite killing. So further, as I told, that degranulation is also another way. So mast cells may also secrete certain antimicrobial substances. So what we did was, so when we incubated them, the pathogens with mast cells, and then we looked for secretion of mediators in the supernatant. So with L. tropica and L. donovani, we did find some significant secretion, but not a lot. But with BCG, there was a very good secretory response. So we saw the secretion of beta-hexosaminidase enzyme as well as histamine. So, and here you can even see that when we performed, SEM, we could see empty granules indicating that, in presence of BCG, they have secreted a large number of contents. So so we can see empty granules here because they have secreted their content. So normally these granules, they are actually electron dense, so they look completely filled. But if they have secreted, then they start looking empty like this. So that is what we had seen. So this shows that they also secrete the mediators. So this is something in which, all the pathogens behaved very similarly. So all of them were showing at least some degree of clearance in presence of mast cells or death. And also ROS production was seen with all of them, and degranulation was also seen to various extents with all the pathogens. And but one more thing after that, so many times, pathogens, to evade the immune response, they also kill the immune cells. So we tried to see what happens. Is there any loss of, viability of mast cells also? And we found that mast cells actually at very high multiplicity of infection with BCG, they start getting killed. Some of them are also killed at about 24 hours. And you can see that also by this release of LDH from these mast cells. So it starts happening, you can say, after 6 hours. So, and when we tried to look for apoptotic death, we found that yes, some of them are dying due to apoptosis also. Though necrotic death here is not very visible, so maybe they are already, so not so much necrotic death, but yeah, apoptotic death is really seen. So similarly, in case of Leishmania also, we saw that the recovery of Leishmania, both strains actually decreases in presence, of mast cells with leishmania decreases. So the recovery of mast cells after incubation with leishmania is lower. And another thing that we saw was that these mast cells are dying and they are also releasing their DNA. So this release of DNA could be because of generation of extracellular traps, because in that case they release their chromatin material. So we try to visualize these mast cells. So this is a form of, you can say, cell death, which is called etosis, which is also a programmed cell death, involves signaling, and it is also mediated by many pathogens. And what happens in this is that this involves ROS production also. So ROS is produced, and that actually then further causes, the whole granule rupture happens, as well as nuclear membrane rupture also happens. And there is mixing of contents of granules as well as nucleus. That means DNA and granular contents are mixed. And then DNA decondensation happens, and the DNA is actually released outside in the form of a trap with great force, so that any pathogens that are there, they are trapped in this. So this is like a spider's web, which is thrown out with force. And since granule contents are also there, so this then contains many antimicrobial substances and enzymes as well. There is a concentration of that, and there are histones there as well. And then they actually ensnare the pathogen. A pathogen cannot run, and also they kill the pathogen there. So this was first observed with neutrophils, but now it is being shown that many immune cells can actually do this. So eosinophils, mast cells, so many of them can form these traps. So here you can see, so these are actually mast cells, control mast cells, but when we incubated them with L. tropica or with L. donovani, then this is DAPI stain. So this is intact nucleus. So you can see that nucleus actually starts to disintegrate in many of them. And these DNA is sent out in protrusions like this. So this is actually, these chromatin structures are very reminiscent of formation of traps. So you can see that more intense traps seem to have formed in response to L. donovani. So further, we performed SEM to look at them in greater detail. And as you can see here, so these are actually control mast cells. And here you can see, again, we can see the formation of these thin structures, so which are the traps. So with BCG also, and also with leishmania, we see the formation of these traps. So one interesting thing that you can see here is, and this we noticed, was that in case of L. tropica, we do not see so many pathogens in the traps. But in case of L. donovani, we can see a lot of pathogens entrapped. So you can see here, all these are actually leishmania. So that means in case of L. donovani, though they are trapped, but they don't seem to be killed. Whereas in case of L. tropica, they are actually killed. That is why we don't see them anymore. So some may be phagocytosed and some are killed. So further, by doing confocal microscopy, what we saw was that here we looked at the extracellular, you can say, portions, and you can see in extracellular portions, we could see DNA, which is stained, blue, along with the pathogen. And also there is histones there is tryptase there. So just as you would see in traps. So same thing was seen even with L. leishmania tropica as well as leishmania donovani. So, and again in leishmania donovani, actually the number of pathogens that was present was very high. So we can see that pathogens remain entrapped, but they are not really killed in case of, especially in case of L. donovani. So further, to see that these traps do play a role in pathogen killing, we treated, mast cells with DNAs. So DNAs will dissolve the traps. So what was seen was that you can see here that when mast cells were incubated, or you can say co-cultured with L. tropica, then they, as shown earlier, there was lots of killing. But when they were treated with DNAs, then a lot of them were staying alive. So fewer were killed. So this showed direct involvement of traps in killing of L. tropica. Actually, similar thing was also seen with L. donovani. Here, though, not so many were killed, but still in presence of DNAs, a lot more were surviving. So this showed that these mast cell extracellular traps were able to kill whatever pathogen was trapped, at least to some extent, though the killing here was anyway not very high. So if we see now comparison of what we have learned from this study, we have seen that mast cells are actually, they are using a multi-pronged, response, or throwing a multi-pronged response to try and catch these pathogens. And pathogens are also in their own way, different pathogens, they try to evade these responses by different mechanisms. So for example, as I said, one of the mechanisms to kill is of course phagocytosis, which happens both for L. tropica as well as for BCG. But in BCG, the problem is, though it is taken up by both phagocytosis as well as macropinocytosis, but at least the phagosome does not seem to be acquiring the LAMP markers, or is not fusing with the lysosomal, you can say, organelles or lysosomes. In case of this L. tropica, they are probably killed inside the phagosome. And as far as L. donovani and L. tropica are concerned, both of them cause formation of traps, and same way BCG also causes formation of traps. And here L. tropica and L. donovani both may be killed, but L. donovani is also resisting killing to some extent, because we see a lot of them, you can say, trapped in the traps, but not really killed. So further, these things need to be seen in greater detail to understand what is happening. So what we are doing now is we are actually trying to do a proteomic and a secretome study of this mast cell leishmania interactions. And further, actually, mast cell BCG interactions also have to be developed, or you can say looked further in great detail, to know exactly how, mast cells are playing a role, and then also how they may be affecting other innate immune responses further by releasing certain mediators. So this is actually our ongoing work. And so if we can actually find that how these pathogens are evading these immune responses, then we can get some new, therapeutic targets for these. And also, if the initial innate response is understood, then, the failure or success of vaccines can also be, ensured. So that is why there is so much, need to study actually how these cells, which are the first responders, how they respond to these various pathogens. So this is what we are actually doing. And so these are some of the publications regarding muscles and BCG in the last few years that we have published, and these are some of the references that I refer to. And I would actually like to thank our collaborators. So Dr. Selva Pandian from Jamia Hamdard Institute of Molecular Medicine, and Dr. Hira Nakashi from FDA USA. So these are the people who introduced me into, Leishmania research, and they are collaborating with us on this. And also Dr. Randhir Day from USA, FDA USA. And I would like to thank your Dr. Paul Roche, uh, NIH, for providing us with these mast cell line. So he is the one who actually introduced me to mast cells, and yeah, I have been hooked since and have been working on mast cells since then. And of course, Professor Rajiv Saxena, who was also my mentor for PhD, and also who provided me with BCG, and that is what I have used. And these are the two students who have performed most of this work. And of course, infrastructure at JNU has been very useful. So all the SEM, TEM that you have seen, all the electron microscopy, confocal microscopy was performed in JNU. And also our central animal facility is also quite good, and they are the ones who provide very good support, especially for immunology research. And of course, all the funding agencies like UGC, DST, etc., ICMR, for providing funds to do this research. And finally, thank you all for your, attention and your patience. So I will take any questions. Thank
It was a wonderful talk, Dr. Niti, and I would like to start with the questions.
you. Yeah, sure.
So, at the end of your talk, you mentioned about a proteomic study that you are trying to do with
Yes.
the mast cells. So I think
Yeah.
this would be very interesting to even study in the context of cancer as well, because inflammation has both activating as well as remission effects in case of
Right.
cancer. And maybe in this context, this kind of a proteomic study would also be useful.
Yes, I agree with you, because so we have one stream of work going on in lab where we are also looking at interaction of mast cells with hematological cancers as well. So, and we are finding very interesting results there as well, which we have recently published, that similar cancers, like we were studying lymphomas, T cell lymphomas. So some T cell lymphomas are actually, killed by mast cells, interaction with mast cells, while others are proliferating. So exactly opposite, effects. So then, though not proteomic study, but we have looked at, upregulation and downregulation of some signaling pathways involved in survival and, apoptosis and those kinds of things. And it was very interesting that in both of them, actually, it was exactly opposite.
Mm-hmm.
Whatever was upregulated in one of them was actually downregulated in the other. So we are now trying to look at receptors and the expression of various receptors on these tumors and those kinds of things. But yeah, as you said, that proteomic study will be very useful, because that will give us immense
And
information.
especially, I think, this is not very well explored. I mean, people have
Right.
shown that, inflammation has positive as
Right.
well as negative impacts, but
Exactly.
really the molecular mechanism is not known. So I think
Right.
it's a very useful thing to study.
Yes. Yes. I agree. Yeah. Definitely. Yeah. Yes.
Thank you.
Thank you so much, ma'am. That was a really good talk. So we have one question from the audience.
Yes.
Ma'am, you talked about L. tropica and L. donovani being trapped
Yes.
into DNA web.
Yes.
Ma'am, so can you put some light why L. tropica is being more killed in these webs as compared to L. donovani?
Yeah, so actually, we have not really looked at that yet. We will be looking at that, but I can answer that from experience of others, from results with neutrophils, for example, because neutrophils also make traps. So they are called as nets, and they also have been shown to trap Leishmania donovani. But what happens there is people have seen that Leishmania donovani is also shown to synthesize a nuclease. So that nuclease can probably dissolve the nets, and that is why they don't stay trapped. And also, there is a special, you can say, the cell, the outer membrane of L. tropica and L. donovani. They are very different. So L. tropica, L. donovani has been shown to be resistant to a lot of, immune, you can say, effectors. So maybe those are some of the things, the phosphoglycans or, lipids that are present there, they are very different. So that could be the reason what makes, L. donovani more virulent in comparison